So it's my pleasure to introduce Dan Knights as our CAP keynote speaker for the first day. So first I'll say thank you everyone for coming. It's been a great first day, I think. So Dan, just to give you a little background on Dan. So Dan is an associate professor of computer science and engineering and in the Biotechnology Institute at University of Minnesota. So he's a colleague of mine. In general, his lab focuses on developing data mining and machine learning methods for analyzing microbiome data and the interaction with the human host. He and his lab has a record of really impactful work in this area. So I think one of the unique things about Dan is that on one hand, he's working on very technical problems. His lab just published an ultra-fast optimal DNA alignment tool. And on the other hand, he's initiating really interesting clinical studies in his lab. So he may talk about a bit today, the immigrant microbiome project, the primate microbiome project. He has a lot of diverse activities that really span the gamut in his lab. I should also mention Dan has a really high bandwidth for being productive. So in his spare time, he started a company. So Core Biome is a microbiome analytics and sequencing company. So in his spare time, he serves as CEO of that company, which is quite impressive. And in general, so it's a real pleasure to have Dan come and as a colleague in our department. Maybe just one sort of fun note at the end here. So many of you might already know this. So Dan is actually the 2003 world champion Rubik's Cube speed solver. So if you happen to catch him in the bar tonight and you have a Rubik's Cube on you, maybe Dan has one in his pocket, I don't know. You should definitely ask him to solve the Rubik's Cube for you. So with that, I'll let Dan take it away. Thanks, Dan. Let's welcome Dan.
Thanks, Chad. Thanks. I am trying to get to the point where I'm better known for my research than the Rubik's Cube. I did a radio interview last year where they didn't ask me anything about the cube the entire time. I kept waiting for it and it didn't come up. And I thought, okay, I made it. So before we start, there we go. I want to disclose a conflict of interest. As Chad mentioned, I started a company a few years ago, which basically provides expert microbiome analysis to industry and academics, anybody who wants it in kind of a commercial setting. But everything I'll talk about today is open source and published. So dinner's coming up. Let's talk about food. I'm going to talk quite a bit about food in this talk and some issues linking it back into the microbiome. You eat this food. Some of you probably ate it today. And I would hazard to guess you actually thought it was delicious too. So look at all these things in this food. Various preservatives, some colors, lots of flavor, chemicals, certain fibers. And in fact, you might even think it's healthy. So I'm gonna, in a little while, tell you what this food is. But the reason why this is important, this is an ingredient list. The reason why this is important is many of these compounds are degraded not by your genes and enzymes, but by your microbial genes. And your microbes are just as interested in what you're eating as you are. And I like to think you are what your microbes eat, because they're basically taking all of those compounds and performing chemical transformations and turning them into various byproducts. The microbiome is the world of microbes that live inside you and has been linked to a wide range of diseases, especially metabolic and autoimmune disorders. But there's probably 100 different major diseases that now have some kind of microbiome signature in them. And in many cases, it's causal. So the microbiome has been causally linked to, for example, diabetes and obesity, which are also rising dramatically in industrialized nations around the world. So this is U.S. Diabetes last 50 years and U.S. obesity over the last or 60 years. And interestingly, these microbiome-related diseases and a lot of the autoimmune disorders are rising largely in the industrialized, westernized countries, not as much in developing nations. And something that you may not know is that people who move to the US actually from a developing nation tend to acquire the US risk of these autoimmune and metabolic disorders. So for example, here's the rate of obesity, BMI over 30, for US immigrants who have been here for 1 year, 1 to 5 years, and so on. And so within just a few years, the risk of acquiring obesity doubles after living in the USA. Here's another study that was on children, and here's obese, overweight, and you can see each of these is roughly doubling over the course of a few years. So there's something changing in the diet and the environment that's causing this very increased risk when people come to the USA. And the first story that I'm going to tell today is called Chapter 1, Born in the USA. This is a study of what does happen to the microbes when people move from one country to another. We know that the risk of these diseases increases. We know the microbiome is related to those diseases. And we know that, that the microbiome is something that could be changing very rapidly. So this is a study that was run by PJ Vange, who was a grad student in my lab. And she is Hmong and was very interested in studying the Hmong population in the Twin Cities. Turns out that there's a very large Hmong population in the Twin Cities. And we ran the study to try to answer what happens to the microbiome when someone moves from here, this is a refugee camp in northern Thailand, to here, the city of Minneapolis or St. Paul. And to do this, we actually collected stool samples from about 550 individuals. And we're going to use these to study the microbiome, what's happening to the gut microbes before, during, after immigration in these populations. And as I tell you this story, we're going to find some interesting things, but it's also going to shine a light on some gaps in what we can do with the current methods. And then we'll talk a little bit about some of the methods development we've been doing to fill those gaps. So to do this study, we enrolled subjects who were living in this refugee camp in northern Thailand, a Karen refugee camp, and then this Hmong village in northern Thailand. The Karen are another ethnic group that have been coming in large numbers to the Twin Cities recently as refugees from Thailand, from Burma. The Hmong started coming to the Twin Cities in the 70s as refugees from the US-led secret war in Laos. And interestingly, when, just as a little aside, when PJ was flying to visit Thailand to collect these samples, she stopped by in Hong Kong and looked up a random Airbnb online. And it turned out to be this guy's Airbnb. He's the CEO of GLBIO, that's running a large biobank of all the Asian microbiomes. So just a little bit of fascinating synchronicity there. And so we managed to get samples from all of these subjects, Hmong, Karen, US controls, and then we sequenced the DNA, and we aligned them to a reference database. And this is how we basically get our kind of primary data product, which is a species table. And the species table can tell us a number of different things. So for every subject, we can say, how much of the microbiome comes from each bacterial strain or each bacterial species. One of the things we like to do is we'll take that species table and turn it into a distance matrix. So now you can see which subjects are more similar to one another, but which subjects are more dissimilar. And then we take that and compress it into an ordination plot so we can project it onto a flat piece of paper and basically see as much variation as possible in 2 dimensions or 3 dimensions. And I'm going to show you that now for the immigration microbiome project. And what you're seeing here is a point for every subject, colored by the ethnic groups and the immigration status. The 2 circles on the left, the 2 ellipses, are the Karen in green and the Hmong that are living in Thailand still. On the right, in gray, we have the US controls. And then everyone in the background are the first generation immigrants. So we see already that there's a complete difference between the people still living in Thailand and the US controls. And then finally, so these first generation immigrants, these are people who moved to the US during their lifetime, and they've been here for anywhere from a few weeks to 30 years. Finally, this last ellipse on the right is second generation immigrants. So we see that as we move from still living in Thailand to first generation to second generation, there seems to be this kind of Westernization of the microbiome. The microbiomes are becoming more and more similar to the US controls. And by the time people are actually born in the US, as second generation immigrants, they're basically overlapping with the US controls. Another thing we can do with this species table is estimate the diversity of each sample. How many species or how broadly they span the tree of life. So that's what I'm going to show you next. This is the phylogenetic diversity. So it's basically what fraction of the tree of life or what distance of the tree of life does your microbiome span. This is the phylogenetic diversity of lean individuals in Thailand. One thing I didn't mention is we stratified the entire study by obesity status. So we have equal numbers of lean and obese in each group. So that's lean in Thailand. Here's obese in Thailand. So even in the Thai group, there's a marked drop in microbiome diversity in people who have obesity. This is first generation lean, first generation obese. So coming to the USA, just moving to the USA in your lifetime is enough, if you're coming from one of these ethnic groups, it's enough to have a significant drop that's larger than the drop associated with obesity. And then if you come to the US and become obese, then you have yet again another loss of microbiome diversity. Second generation obese. So this is the first time that we've actually seen this sort of compounded loss of microbial diversity in a multi-generational cohort that are being exposed to industrialized society. So there's a theory about this called the disappearing microbiota hypothesis, that basically if each generation can only pass on a subset of their parents' microbes to the next one because of antibiotics, loss of fiber in the diet, things like that, then you have this kind of step function where one generation loses diversity in the microbiome, passes on a subset of microbes. The next generation loses more, passes on a subset, and we keep losing more and more ancestral microbes over time. And that's actually what we see implied here. And by the time you get to second generation, they're right about on par with the US controls. There's a lot more in these data that I'm not going to go into right now because I want to have a little bit of time to talk about some methods. But this is the overall finding that, yes, coming to the US is associated with a dramatic shift in the microbiome. You get Americanized in the microbiome, and there's this dramatic loss of diversity. However, these samples are all studied with a low resolution type of microbiome analysis. If some of you may have done some microbiome analysis, if you have or you're familiar with it, this is sequencing this one marker gene. So it's cheap, it's fast, it's very robust, but it's super noisy and it's not very high resolution. So you can't really measure at the strain level, you can't really measure what functions or enzymes are changing. So 550 subjects done using this cheap method. We followed up with the more high resolution, expensive method. This is shotgun sequencing on 55 subjects. And I'm going to show you them here. This is now, instead of looking at the species table, this is looking at the enzymes. So what enzymes are contained in the microbes that live in the guts of these individuals? And what we saw is that going from Hmong Thai to Hmong first, this is first generation, or to the US controls, this is just one of the things we found, is that there were certain enzymes that were quite prevalent in the Thai residents but then went completely missing, or almost completely missing in the U.S.A. And some of these seem to be associated with certain components of the foods that people might be eating in Southeast Asia. For example, there's beta-glucan, which could be targeting these glycoside hydrolases, so that could just be dietary fiber. Some other glycoside hydrolases that are targeting cellulose. Beta-mannan, which is common in palm, coconut, konjac. These are very commonly eaten foods in Thailand and Southeast Asia, and possibly xyloglucan, which is prevalent in tamarind, also commonly eaten in Southeast Asia. So it seems like a sort of use it or lose it type of phenomenon. You come to the US, if you're not getting these dietary components on a regular basis, you may lose the microbes that can degrade them, which, you know, the consequences of which we don't really know yet. So I mentioned that these were the expensive, fancy, high-resolution type of data, shotgun sequencing. Why can't we just do shotgun sequencing on all of the samples? So I told you we had this cheap version on 550 subjects, and then this expensive whole genome shotgun sequencing on just a subset. And this is how the majority of the microbiome studies have been taking place to date. You do the cheap version on all your samples, and then you go do the expensive version on the subset. And something that we've been working on in my lab, I get kind of frustrated by this because I love big data, but I'd like to have good data and big data at the same time. And what we've been working on is trying to fill in something here. How can we get more resolution without resorting to this single marker gene, but without also having to pay for this kind of very deep whole genome shotgun sequencing? And it turns out there are two steps you need to take in order to make shotgun sequencing affordable. One of them is improving the information you extract from the sequencing data. The more information you can extract per read, the less data you need, fewer reads you need. And then you also need to cut costs on the wet lab side. I don't know how to do this, but I have friends who do. And then I worked on this first part. So this is what we're going to talk about in chapter 2, fast and spurious. So the vast majority of all of the alignments that are made are done using approximate aligners. So you've got some input DNA. This could be stool DNA. It could be some other type of environmental DNA. And then the goal is to search it against every strain in a database. There might be tens of thousands of strains. We saw it in the last talk. And ideally, we'd like to find the best match every time. But that's not necessarily feasible. So it's feasible for small data sets due to this wonderful algorithm, Needleman-Wunsch algorithm. And you've probably all studied this at some point in your academic careers, that you basically have to fill in this table. It's a dynamic programming problem where you fill in this table row by row. Every row is a base in the input sequence, the query sequence, and every column is a base in the reference sequence. In practice, these are somewhere around 100 bases long, and these might be 5 million bases long for a bacterial genome. And the goal is you fill in the table row by row, keeping track of these scores, and then you find the best score at the bottom and sort of trace it back through the table. And that tells you the optimal gapped alignment of the query to the reference. So where you have to basically slide it along the reference at every possible point and insert gaps at any possible point to find the best match. That's the challenge. And to do this on an entire shotgun data set would be very time consuming. So you might have about 10 billion query sequences. These are the shotgun sequences. 100 bases each times, say, 10,000 references times 5 million bases per reference. It's about 5 times 10 to the 23rd cells you have to fill if you wanted to just brute force your way through and get the optimal alignment for the whole data set. This actually is just about 1,000 times more complex than Rubik's Cube. I always like to have a cube in my talk somewhere. So a cube is 4.3 times 10 to the 19th. So sorry, it's actually 10,000 times more complex than finding all the states of Rubik's Cube, which took 35 years of CPU time on Google's cloud computers to do. So here's something, a practical way of looking at this problem. If you had a single shotgun sequencing sample, and you want to use the Needleman-Wunsch algorithm to compare it to the entire strain reference database, it would take about 4 years, just using this naive algorithm. There have been efforts to speed this up over the next few decades, and some of them have been quite good at parallelizing different parts of it, including, this is one of the leading ones, GL Search, which is part of the FASTA toolkit, which had an extraordinary speed up, got the speed down to about 75 days. But this is on a supercomputer, so still too slow to actually run on a real data set. And we were sort of working on making a better approximate aligner when we stumbled across some optimizations that we hadn't really thought of before. This is work that was done by Gabe Algaleef, who was a grad student in my lab, who's now a postdoc at the Janssen Microbiome Institute in Boston. What he realized was, and this wasn't on its own wasn't new, that as you got further and further into the table, you could actually prune off whole sections of the table that you could prove couldn't reach the optimal solution because the score was too far away from optimal. So even if you had a perfect alignment from then on, you couldn't reach the optimal score. So that's great for, by doing this, you can get about a 2x speed up for free. So you can prune the bottom half of the table. Now you're down from 75 days to 30 or 40 days, still too slow. Then he realized that if you sort the input sequences alphabetically, and you just cache the results from the previous sequence, the results for the first half of the table are the same as the previous sequence. So you can basically prune the entire first half of the table. And these are random shotgun sequences, so on average, you might have about half of the sequence be the same as the previous sequence once you get up to hundreds of millions of random sequences. So now you can guess what he did. He took the two optimizations, overlaid them, and in practice what happens is that although on any individual search, it's still slow, you still have to fill the whole table, when you have millions and millions of sequences, you can take advantage of this additional pruning step, and you basically never have to do the whole table. In fact, you hardly have to do any of the table most of the time. So the search becomes extremely fast, and we can now do the same samples in about 5 minutes, assuming that you're processing them all at the same time. So what does this mean? It means that we need less sequencing to get the same signal. So we're now doing optimal alignment on full shotgun data sets. Every read that's a marker, we find it. We never have spurious hits to the wrong strain. And so we get optimal precision and recall because it's a full gapped alignment. So the other steps I mentioned, optimizing some of the wet lab protocols to remove some of the library preparation costs. For those I have to thank Kenny Beckman and Daryl Gold at the University of Minnesota Genomics Center. So they did some great work cutting costs down on that side. And we were able to roll out an approach to shallow shotgun sequencing that's now under $100. And that's when in the commercial setting, if you have your own sequencing core, which are sometimes subsidized, you can get the cost closer down to $50. So it's almost the same cost as doing the cheap, noisy 16S sequencing. And now, so we have this new tool where instead of having to choose between noisy big data, or high resolution small data, we can actually run shotgun on a whole data set, very large data set. And I'm now going to show you an application to one study where we did this. So I told you we were going to talk about food. This is called Chapter 3. Don't just read the label. So this is work that was led by Abby Johnson, who's a postdoc in the lab. And Abby has an interesting background. She has a PhD in nutrition at the University of Minnesota. Then she went to Nestle and worked in research at Nestle for a while, and then decided to come back to the university and learn how to do microbiome analysis. At the same time that I was saying, gee, I really wish I had someone who knew nutrition so that we could develop better nutrition methods. So she's been in the lab doing work to basically improve how we analyze diet data. And she carried out this study as part of that development work. So this is a study where we used these new shallow shotgun sequencing techniques to do shotgun sequencing every day for 2.5 weeks on 34 individuals. And that's what I'm showing you here is grouped by individual, the time series of their microbiome. And because it's shotgun sequencing, we have species level resolution, and we also have functional resolution on what the enzymes are. So this is a zoom in on Abby's microbiome. She had one of the most stable microbiomes, which she was very proud of. And we have, you can see every day sampled her microbiome. You can see here some other things. Each person has their own kind of fingerprint. Some people have some neighbors, like these 3 people on the end, they all are Prevotella carriers. That tends to be a non-US bug. That's a bug that's more prevalent in developing nations. So these people have somewhat similar microbiomes, although if you look at the rest of the bugs, they have some differences. Everyone tends to maintain their fingerprint over time with varying amounts of variability. So some people are more variable than others. And importantly, we were able to get all of this very high resolution data at the same cost as a single time point of deep shotgun sequencing. So of course, I mentioned Abby came from a nutrition background. So we collected these data along with nutrition data. And nutrition data are really exciting. It's been really fun to work on these. This is the highest level of food group, very basic, like dairy product, grain products. And already you can see it's so much more variable even than the microbiome. Still individualized, everyone has their own kind of diet fingerprint, but it's more variable than the microbiome, with a couple of major exceptions. What do you think happened to these people? Well, what Abby told me is that this is what happens when you do research with computer scientists, is that these 2 individuals were drinking nothing but Soylent the entire time. Nothing but Soylent the entire 17 days of the study. So they provided sort of an interesting sub-study because their microbiomes did not stabilize. Interestingly, a monotonous diet does not stabilize your microbiome. We excluded them from a lot of the analyses. And then what we did is we started to look at how does the microbiome relate to the foods you're eating? And this is where we get to that nutrient label, because the diet, as I said, is more variable than the microbiome. It's highly personalized. It's very sparse. It's noisy. So actually, as I said, a very exciting area for data science, I think. How are we going to represent this in a way that we can integrate it with the microbiome data and better understand what's happening in healthy populations, populations with obesity, populations like the immigrants that we studied earlier. Most studies on the microbiome that have collected diet information use the nutrients from the nutrition label. So there's about 100 nutrients that are known for most common foods. And they're, many of them are very broad things like carbohydrates or sugar. But of course, sugar has many types of sugars. And the microbes don't just care about sugar. They want to know what kind is it, because they may or may not be able to degrade it. So we started off with nutrition data. And what we did is we took, for every person, we took their average nutrient intake over the whole study, 17 days, just collapsed it down to one time point. We took their average microbiome over the whole 17 days, collapsed it down to one time point. And then we made these ordinations of them and tried to overlay them to see if they would line up. So basically, if you're similar to someone in diet space, are you also similar to them in microbiome space? The answer was no. at least not when we looked at nutrients. We did not get a significant correspondence between the nutrient distances and the microbiome distances. So we started to think about this a little bit more. And this actually kind of made sense because remember this mystery food we were talking about earlier? This has all of these other ingredients that are not listed on the nutrition labels that the microbiome cares about deeply. And we're not capturing those in the nutrient data. So remember this food I mentioned that you all probably like to eat. This is the food. Those are the ingredients of a strawberry. And this, because it's considered sort of a whole food on its own, this is the nutrition label for a strawberry. So that's what the FDA requires you to put, on a label of a strawberry. And I have to thank Robin Shields Cutler, who was a postdoc in my lab for the design work, and also James Kennedy is a chemist in Australia who had the idea for doing this and was really kind and let us modify his work. So, we're stuck because most of the studies are using these nutrient labels. We don't have this information for the vast majority of foods. I'm not going to go into the details now, but we worked on some methods where we could basically share information from closely related foods to get more statistical power out of the diet data. And then we were able to link that back to the microbiome. So this is, if you're eating similar foods to someone, so it's still diet distances overlaid on microbiome distances, but here the diet distances are determined by how closely related our foods are. So if one person eats kale and spinach, one person eats kale, another person eats spinach, their diets will be more similar than someone who eats an Oreo cookie. And when we use those distances, now all of a sudden we get a significant correspondence with the microbiome. So, and interestingly, the way that we did this was to use a phylogeny of the food. So we were able to take some methods from the microbiome field that leverages phylogenies for comparing microbiome profiles. We used them on diet and we were able to find this correspondence. But because we had this dense species level data, we were able to actually zoom in. So remember, these were 1 point per person. So this is the person's average diet collapse across all 17 days, average microbiome collapse across all 17 days. We were able to zoom in and look at the actual diet microbiome associations in each person over time. So as you're changing this food, what's happening to the microbes in your own microbiome. And what we saw is that there were a few that were quite generalized. So some of these cookies and cakes and bars that tend to have a lot of chicory root or inulin fiber supplementation tended to be positively correlated with Roseburia inulinivorans, a bug that likes to degrade inulin in a number of individuals. But there were other associations, such as vegetables, dark green vegetables, being negatively associated with this microbe in some people and positively associated in others, and pork being negatively associated with that microbe in some people and positively associated in others. So the story is much more complex than we had anticipated, in that it does seem to be that the food choices do predict what's going to happen to your microbiome, but in a personalized way. And in fact, we verified this, that if you train a model in your own dietary history and you tell the model, what does your microbiome look like today? What did you eat yesterday? You can predict what your microbiome is going to look like the next day. If you take a model trained on someone else's data, it doesn't work. So, so this kind of demonstrates that we need more studies that are longitudinal. We need more large studies across many different people with different microbiomes and different diets and able to understand this. So in closing, I've talked about a few different points here. The first one was the Immigrant Microbiome Project, which was basically demonstrating that industrialization seems to be causing this loss of ancestral gut microbes that are related to many diseases that are at the same time on the rise in industrialized nations. Then, I talked about how we have some methodological issues that were making it hard for us to get high resolution on those data sets. We think that dietary interventions might help treat microbiome-related disorders because what you eat plus your microbiome does predict what's going to happen to your microbiome the next day. But the microbiome interactions with diet are very complex and they seem to be personalized. So we need large studies and we need dense studies. And then finally, I talked about some new methods to make shotgun sequencing more affordable that will, I hope, enable people to do large and high resolution diet microbiome studies for the next phase of microbiome research. And I recommend trying that tool out, the burst aligner. It's really fun. There are lots of times when I use it, even in just research questions. It's very satisfying to know that you're getting not just the best hit, but you can also return every optimal hit from a data set. So if there are ties, you'll find them. So I recommend trying that tool out. Finally, I'm hiring bioinformaticians both in my academic lab and at CoreBiome, depending on your interests. So please contact me if you're just graduating from your PhD program and are looking for something to do next. And I'd like to thank the organizers for inviting me here. Thank you for your attention. And thanks also to the members of my lab and the many people who've helped out on this work. And I'd be happy to stop there and take any questions.
So we have time for some questions. Right. Oh, yes.
So where did you get the information about what's
The strawberry is one that's been studied really well. And so there are a few foods like that where we have a near complete list of every chemical component. People have done mass spectrometry on it. But for the majority of foods, we just don't have.
Is that compiled somewhere, or you have to sort of dig through the literature?
That was work that James Kennedy did. He was the one that I cited for the picture, and he makes these really cool posters where he's someone, he talks a lot about chemophobia and tries to convince people not to be afraid of chemicals. And so he makes these cool posters that say, all natural strawberry. And then it's got all the nasty looking ingredients. So he was the one who compiled all of that from the literature.
Got it, okay. It wasn't that he read all the spec, but he pulled the information out.
No, that's been done over many years, yeah. And there is work going on to try to characterize more and more foods using mass spec. But even then, how you prepare the food changes the mixture of components.
Sure, because you might break something down.
You break things down, and where it's sourced from changes the components. The strain of the corn or whatever changes the components. Yes?
Could you speak to the biology of food and sort of how you went about compiling that?
Sure. The phylogeny of foods, which we usually work really hard on naming all of our tools. This one we called it Food Tree. What happened was Abby was, and there's Abby in the middle there. Abby was looking through the U.S.A. food database that our dietary questionnaires were linked to, and she noticed that the food codes had hierarchical structure to them. And so there was, dairy starts with 1, and then fermented dairy starts with 1, 0, and so on down until you get to the actual food. So it essentially was basically crowdsourced from 100 years of nutrition experts manually entering every food into the right food code.
Fascinating stuff. So the outcomes that you're studying, specifically, let's say obesity, have also been linked to truly environmental exposure. So pollution of air, water. You're looking at the microbiome. So I've always wondered what, if any, is the relationship between the truly outside environment and the microbiome that you profile from the gut of other places.
That's a great question, and it's confounded in our studies, and that's a tricky part. So that's something that we're trying to follow up on now in mouse experiments. we have this mouse experiment underway at a Mayo Clinic with my collaborator there, Prana Kashyap, where we're basically going to have at one end of the hall, like mice in a little cage with like a miniature jungle and a waterfall and a, you know, and then the far end of the hall is going to be like a miniature skyscraper with smog and honking horns and stuff. And basically try to simulate, you know, not exactly that, but simulate all of the different insults that can happen to the body and the microbiome in industrialized society to find out which ones are actually having the largest effect on the phenotype and the largest effect on the microbiome. And if we can find something there, then we can sort of dissect and learn more about that one aspect.
Here's a question up in the middle there, and then we'll take one down. Go ahead.
Just to expand on this question, sort of related to that, the more pharmacological agents taken by people, more sterilized environment, maybe acting, you know, they've got microbiome as well. Since we are American politics doing antibiotics as well as have access to better medical care. At least the data that we were seeing at the beginning, the general reduction diversity microbiome.
Mm-hmm.
I was wondering if you guys went to pretty obvious as far as how you guys went about avoiding the confounding factors, or even looked into what kind of medications people in your study.
Mm-hmm. So pretty much every study we run, recent use of antibiotics is an exclusion criterion. And so, you know, same for that study. But we don't really have a controlled way of telling how many times a person's been exposed to antibiotics throughout their life in that group. Antibiotics are actually quite common in many developing nations, sometimes even more common than in the U.S. But we don't have information on sort of like their cumulative exposure. One thing that I think is potentially a factor is not antibacterial antibiotics, but antifungal or antiparasitic antibiotics, because a lot of, especially refugee groups who come to the U.S., they get exposed to antiparasitics very early on, which could thoroughly disrupt the microbiome, you know, the sort of stasis that's been established between parasites, you know, eukaryotic parasites and bacterial symbionts. So I think that's a factor that we don't have measured that might be influencing it. And I think you also mentioned hygiene, which that might also be it, and that it's really hard to study in the human population. But there's so many things confounded. There's stress, there's antibiotic exposure, antiparasitics, food sourcing, what are the microbes on the food? There's the water supply, could be the chlorination in the water, hygiene, household crowding.
Yeah, I understand that.
Yeah. No, there are a lot of, we just don't know. We don't know yet. That's why we're trying to go into the mouse experiment and do antibiotics on their own, antiparasitics on their own, and so on.
So down here.
Yes, thank you. The same way that you have done a study about the microbiome and its change over time of the immigrant population between the first generation, second, and so forth. There are also established studies that show the same type of pattern happens with people who get cancer, whether it's prostate cancer, breast cancer, and so forth. But by the time they approach the family approaches the second generation, they're at the same risk level as a local population that's not a microbe. Have you ever had any type of a correlation or looked at the parallel between your study and that? And as a matter of fact, in the established studies for cancer, diet has been a prevalent cause of the increased risk for that.
We haven't looked at that, but I'd love to chat with you about, you know, if you have some ideas on references for that.
Yeah, I'll be happy to send you some.
Okay.
I can get your email.
Great, thank you.
Let's take one last question down here. Yeah.
So as you mentioned that moving to US decreases the diversity, but what happens if somebody goes back to Thailand, let's say, as a tourist or for a short time, will it increase decrease the diversity?
We don't have that data yet. I'm trying to get Jonathan Krive, he's the CEO of Asia Biome, and he travels all over the world and stays each place for a month. And he's agreed to sample himself for an entire year. I'm just going to provide the kits and the sequencing, trying to figure out how to do that. But yeah, we don't really have any data on someone going back for a long period of time, either from from industrialized nation to a developing nation, or someone who came from developing originally going back. We don't have those data yet.
Okay, let's thank Dan again.
Thank you.